Hi guys, welcome to my channel. Today we'll be doing the chapter from which every pre-med starts their MDCAT journey from, and you guessed it right, it's enzymes. An easy chapter, but still we have a lot of content to cover. So let's get started. What are enzymes? Let's move on with the definition first. Enzymes are biocatalysts that speed up a chemical reaction without being consumed in the chemical reaction itself. Okay? And how do they speed up a chemical reaction? They do so by lowering the activation energy. Now the question comes, what is meant by activation energy? So the activation energy is the minimum amount of energy required to start a chemical reaction. Uh, so for example, we have this graph here, right? This is the energy of products. This is the energy of reactants. And now this energy here from converting from reactants to product is the activation energy, not this can be done on its own, but this green highlighted one is the activation energy required to convert the reactants into products. And the enzyme speed up a chemical reaction by lowering the activation energy. You can see it in these graphs here. This activation energy when the enzyme is added is much lesser than this one, right? So we can say this is a catalyzed one. Okay, wait. This is the catalyzed reaction, right? I can also highlight it is the minimum energy required to start a chemical reaction. Also, in this definition here, we have that enzymes are biocatalysts that speed up a biochemical reaction without being consumed. And if they're not consumed, we can say that they can be used again and again, right? Also, that means they're required in very small amounts because they can be used again and again. So if one enzyme does its action, then it can be used again for the same reaction. Also, we have to know that enzymes can work in vivo, that is, in living cells, as well as out of living cells, that is, in vitro. Uh, an example of uh, uh, an example of enzyme working in vitro is, for example, we have tag polymerase, which is used in which you'll also read about in biotechnology, but it is used in PCR, polymerase chain reaction, and we do not do it inside living cells, we do it outside of living cells, right? Okay, moving on, we all know that all enzymes are globular proteins. And what does it mean by globular proteins? It means that they're water loving, that is also hydrophilic in nature. Also, it means that they're spherical in shape, right? Except there's an exception here. You have to remember everywhere, wherever exceptions comes in biology, you need to remember what it is because MCQs are based on that. Except ribozymes, except ribozymes, they consist of RNA and are found in ribosomes. Right. Moving on to the structure of the enzyme, we have the active site here, we have the allosteric site here, the substrate which is complementary to the active site is drawn here, and there's also cofactor which we'll read about further. First of all, starting with the active site, we can say what is an active site. It is a specific charge bearing site, and it is a 3D cavity in which the substrate binds, right? Specific, you have to highlight a specific and a 3D cavity. Okay, and it consists of 3 to 12 amino acids. Also, it can be written in some books that consist of a small amount of amino acids. That is also correct, but sometimes they also ask the exact amount of amino acid that it is made up of. So it's 3 to 12 amino acids, right? Active site consist of two types of sites. First is the uh, first is the binding site. It is made up of two sides. First is the binding side, and the second is the catalytic side. So what is meant by the binding site? It is uh, considerably less specific than the catalytic side. And why is it less specific? We'll see that in the part where we do inhibitors of enzymes, right? Also, it chooses the substrate. It doesn't do any catalytic function. It just chooses the substrate which is complementary to the active site. And in this case, for example, I can give you a scenario here just so you can remember why is it less specific. Is that for example, there's this one person here. It's a very non random example, but you can remember it easily. For example, there's a one person here. This guy likes another person here, right? But the problem is uh, likes this person here. But the problem is this person has a twin. So they are similar, right? They're similar, but they're not the same. So for example, if this is the binding side, for example, if he sees this person instead of this person which he actually likes, he'll be like he can add that to the enzyme as well, because he just chooses the substrate which is complementary, and this can also be complementary to the enzyme. Maybe the shape, for example, this person, wait, let me show you. This person right here has this shape to the complementary to the active side, and this is the enzyme, right? And now this twin of her or whatever has, wait, where did my, okay, this twin of her has this shape, and this is also the enzyme. Now the enzyme is confused between this which substrate we are supposed to use. Okay, so the site is therefore less specific. Let me clear that out. It does not look good. Okay. And why the catalytic site is more specific? Because it performs the catalytic activity. It converts the substrate into products. And now if the binding site has not chosen a correct substrate, that means it cannot be converted into product. That it will just remain binded to that, but it will not be converted into product, right? Okay, moving on to the cofactor. So what a cofactor is, it is a non-protein part of an enzyme, and it is required for a proper functioning of some of the enzymes. So you need to remember that not all enzymes need a cofactor to work. And also you need to know that if, for example, you can say that the cofactor is is not present in the enzyme. For example, this enzyme is without the cofactor, right? And this enzyme needed a cofactor, right? This is not the enzyme that does not need a cofactor. This one needed a cofactor, and without the cofactor, what it is known? It is known as an apoenzyme. I'm writing it down here. It is known as apoenzyme, which is basically an inactive type of enzyme. Without the cofactor, it is inactive because it already needed a cofactor. That does not mean that it is without. If this enzyme did not need a cofactor, it will not be known as inactive enzyme, right? Also, if for example, now the cofactor is attached to it. Okay, where did my cofactor go? Okay, now we'll say, I don't know why my cofactor is going there. Okay, now we can say, like, for example, now this cofactor is attached. Now this enzyme will be known as holoenzyme. Wait, holoenzyme. I can write it down here as well. Enzyme without cofactor is known as apoenzyme, and with, I'm a lazy person, okay, with cofactor it is known as holoenzyme. Okay, so this enzyme is the protein part and the cofactor is the non-protein part. Moving on, we need to categorize the cofactors. There are two types of cofactors. One is the organic cofactor and the other is inorganic cofactor. So what does it mean? For organic cofactors, we have another two type of cofactors, which is one is the coenzyme and one is the prosthetic group. So I'm sure most of you do know, which are who are from A levels know what are coenzymes. Coenzymes, an example of them include NAD, FAD, and ATP. Also, we need to remember that they're the derivatives of vitamins. And also in inorganic cofactors, we have the activators. Easy to remember because, oh my god, what was that color? Okay. For an inactivator, example of inactivator are usually metal ions. For example, zinc 2 plus, Mg 2 plus. Now we need to remember which are a covalently bonded cofactor, which are like detachable cofactors. So an easy way to remember is that only prosthetic group is covalently bonded. The rest, the coenzyme and the activator, are both detachable cofactors. Means they can be removed from the enzyme and the enzyme can become apoenzyme. But if we have a prosthetic group, they are covalently bonded, very strongly bonded, not easy to remove from the enzyme, right? Let me remove this color, it's giving me something. Okay. Okay, moving on the mechanism of enzyme action. Everybody knows how an enzyme works. First of all, the active site chooses the substrate which is complementary to the active site, right? This forms the enzyme substrate complex. After the enzyme substrate complex, the substrate is converted to products while being attached to, this is the key, while being attached to the enzyme. So the enzyme product complex is formed because the substrate has not been removed from the active site and it is converted into the products while being attached to the enzyme. The product is released but the enzyme remain unchanged. This is the keyword here again so that it can be used again and again as we saw earlier. Now, moving on to the important part, this one is more important because two mechanism of enzyme action were introduced. One was the lock and key model and one was the induced fit model. First of all, we'll see the lock and key model and then we'll move on to the induced fit model. So the lock and key model was introduced by Emil Fischer in 1894. However, there are some problems with the our provincial books. Like in PTB, we have it was introduced in 1890, and in STB, which is the synth textbook, we have 1898. So you need to remember the date according to your province books. Okay. The lock and key. What does the lock and key model says? It lock and key model says the active site has definite shape. It can is not flexible. It can only bind one type of substrate. That means it can bind only one type of substrate and it can carry out only one particular reaction with that substrate only, right? Because it has definite shape which will be complementary to this substrate only, to its own type of substrate, and it is very fixed, very loyal to the substrate, we can say. And also the enzymes which follow the lock and key mechanism are also known as non-regulatory enzymes. The examples of them are sucrase and maltase. Sucrase will work on sucrose. I write it down here as wait. it will work on sucrose, and maltase will work on maltose. Okay. Moving on to the induced fit model. Induced fit model was introduced by Koshland in 1959. There is uh, no competition with this. No changes of date in different provincial books. So you can expect this date to come rather than the 1819 and 1898 one, because this one is a definite one. Okay. Also, the induced fit model says that active site is flexible. The key word here is that it molds itself around the substrate. Uh, it molds itself around the substrate. For example, we have a substrate which is not exactly complementary. We can we see it here as well. We have an active site here, and the enzyme is, and the substrate is not exactly very complementary to this enzyme. So we can say that the enzyme can change its shapes. The enzyme active site can change its shape, and it can become complementary to this substrate. Okay. And because it is flexible, it can carry out more than one type of related reaction. Not that it can carry out all the reaction with all the substrate. That does not mean that. Okay, it can carry out more than one type of reaction. That is true, but it can carry out only related reactions. Moving on, the enzymes which work under the induced fit model are known as regulatory or allosteric enzyme. This is important because this is a straight up key point, right? Regulatory or allosteric and non-regulatory enzyme. You need to remember this. Then the enzyme which comes under the induced fit model are the carbonic anhydrase, which can work on both CO2 and oxygen. We'll see that in the life processes that we do. And also hexokinase, also an induced fit model mechanism following enzyme. Okay. Now moving on to the factors in affecting enzyme activity. First of all, we'll do the very OG one, which is temperature. In temperature, you need to remember three types of temperature. First of all, we'll see the graph, and then we'll move on which three types of temperature we need to know. So we can see, as the temperature is increasing, enzyme activity is increasing. It reaches the max here, and it reaches the max at the optimum temperature. The temperature at which the enzyme reaches its maximum activity is known as the optimum temperature. Okay, now we can see after the optimum temperature, as we increase the enzyme activity, as we increases the temperature more, the enzyme activity decreases until it reaches zero. Right? We can say there at low temperature. At low temperature, enzymes become inactive. We can say they become inactive. That does not mean that they become that they are destroyed. Okay, inactive does not mean that they're destroyed. Increasing the temperature again will make them active again. Okay, they are not denatured, as we say. Okay, so the temperature which is used to convert an inactive enzyme to an active enzyme is known as minimum temperature. You need to remember this. You needed to remember optimum temperature. You need to remember minimum temperature. Again, what is minimum temperature? It is the temperature at which the enzyme is converted from inactive to active form. And why had it become inactive in the first place? Because we have lowered the temperature, and that does not mean we have destroyed the enzyme. Increasing the temperature will make it functional again. Okay, but that is not the case with high temperature. At high temperature, enzyme becomes denatured. That is, it cannot function anymore. And the temperature at which the enzyme cannot function anymore is the maximum temperature, because at that temperature it has become denatured. It has lost its active site. It is not complementary to the substrate anymore. Okay, these are the two type of optimum temperature you need to remember. One is for humans and one is for birds. Humans have an optimum temperature of 37 degrees centigrade, and birds have an optimum temperature of 41 degrees centigrade to, I think, 43 degrees centigrade. Some books have written 43 degrees centigrade as well, but it's okay to remember this one value, and you can choose the MCQ with regards to that. Also, there is this one more point that you need to remember, that increase in 10 degrees centigrade of temperature doubles the rate of enzyme activity. The key point here is doubles the rate of enzyme activity and increase in 10 degrees centigrade of temperature. Okay, moving on to the pH. Now let's look at the graph first, and then we'll see what it is about. We can see that this is the point, the green highlighter one is the point where the enzyme activity, we are supposed to have enzyme activity here, okay? Enzyme activity is maximum at the optimum pH, right? And uh, mm, below and further from the optimum pH, in both cases, the enzyme activity is decreasing. So we can say slight change in pH causes ionization of amino acid at the active site. Okay, causes slight changes in pH causes ionization of amino acids at the active site. That is not as severe as when there is extreme changes in pH. When there is extreme changes in pH, this disrupts the ionic bonding, and when the ionic bonding is disturbed, the function of the active site is lost. Okay. So also there's this table in all the books, which is very important. It is of enzyme against optimum pH. You have to remember these enzymes and the optimum pH. You can see, either you can remember this entire table, or you can just see through it like twice or thrice and then attempt the MCQs. And then some people also remember the values by attempting the MCQs rather than memorizing the whole table. Also, there's this mnemonic which is very helpful. You guys can easily remember this one, and it resembles that this is pepsin, this is pappu, sucrase, say enterokinase, ethine, salivary amylase, C, catalase, catalytic, chemotrypsin, chemistry, pancreatic lipase, parhi, or arginase, arch. And it's very easy to remember these enzymes in order with this mnemonic, and then you can just remember 2, 4.5, 5.5, and you can remember 6.5, 7.5, and you can remember it like that. Okay, moving on to the our third factor which affects enzyme activity is the substrate concentration. We can see, let's look at the graph. We can see as the substrate concentration is increasing, enzyme activity is increasing until it reaches this value of substrate concentration at which enzyme activity becomes constant. It plateaus, the graph plateaus off. So the enzymes are no longer functioning. I'm sorry, that is not the case. The active site of enzymes are saturated. That means at this site, at this point in graph, the active site of enzymes are saturated. Enzymes are working at their maximum capacity. Enzymes are working at their max capacity. All the active sites are filled. Okay, whatever the amount. For example, I can give you a value. For example, we have three enzymes here, right? We have three enzymes here. These are the same type of enzymes. Please ignore my drawing. Okay, we have these substrate here. Now, first of all, we had two substrates. Increasing the substrate concentration, we have introduced another substrate. These are all same. Okay, please ignore my drawing again. Okay, see. So first of all, we had two type of substrate. Let's see. First of all, we had two type of substrate, and we have three enzymes, right? So the enzyme, one active site is still left. So if we increase the substrate concentration, that is add one more enzyme, then the activity of the enzyme will increase, right? But if we add here one more substrate, now we do not have an enzyme for it to act on. That means all the active sites will be filled, one by this, one by this, one by this. Okay, so now this enzyme substrate is left. What if we add another substrate? What if we add another substrate? The case will remain the same. All the active sites are filled. The enzymes cannot work anymore. So as I was saying, the enzymes cannot work any more than that. There are already three enzymes. There are already three enzymes. They are using three substrates, and these three substrates are extra. They cannot work any more than that. So that means the active sites are saturated, and enzymes are working at their maximum capacity. Okay, removing this so that this does not come in the PDF. Okay, moving on, we have inhibition. There are two types of inhibition. One is competitive inhibition, another is non-competitive inhibition. In competitive inhibition, what the substrate, the inhibitor is similar to the substrate. Now we need to remember that inhibitor is similar to the substrate, and that does not mean that it is the same as the substrate. It is the similar case of that twins. The twins are similar, they're not exactly the same, right? Something must be different in that. Okay, so if the inhibitor is similar to the substrate, that means inhibitor can bind to the active site. That means it can be complementary to the active site, and the binding site, which is less specific, chooses the substrate, thinking that it chooses this inhibitor, thinking that it was a substrate. Okay, however, the competitive inhibition is reversible inhibition. Um, it can be reversed by increasing substrate concentration. For example, I can show you here, right? I can show you here. We have this enzyme, okay, and this is the uh, original substrate, right? Okay, and this is the inhibitor. Okay, so now if we have introduced more original substrates in the solution, these are all same, okay, and there is less, only one inhibitor here, what is the chance of the inhibitor binding to the active site? It is less, right? So if we increase the substrate concentration, the chances of inhibitor getting attached to the active site is less. That means it can be reversed. The competitive inhibition can be reversed, and the enzyme function can return again. The example which is associated with this is penicillin antibiotic, which is a competitive inhibitor. Moving on to the non-competitive inhibitor, the inhibitor in this case is not similar to the active site, to the substrate. Okay, right? Sorry, it is not similar to the substrate. It does not resemble the substrate in any case. Okay, does not resemble. This type of inhibition is irreversible. Also, the inhibitor binds to the allosteric site. It was when we saw the structure of an enzyme, we saw that the enzyme had an allosteric site as well, right? So this was the active site, and this one here, in which the substrate does not bind, that is meant by that is the allosteric site. And when also, okay, this is the allosteric site, right? And when there, for example, there is a inhibitor here, this is the allosteric inhibitor, it binds to this site, it changes the shape of the active site. That is how it works. Okay, so the enzyme active site will not remain this, it will get converted into this, for example. Now the substrate, which was originally this, cannot bind to the active site anymore. That means that the enzyme is totally destroyed, it is irreversible, this cannot detach easily, and it is very strongly bonded. That means the active site remains disturbed, and the substrate cannot bind to the active site. Okay, so that means it binds to the allosteric site. The example of this is DDT, which is a banned pesticide. Okay, now these are the graph of inhibitions. First of all is the uninhibited one. This is a normal enzyme activity against substrate concentration. For example, this is the original graph. In competitive inhibition, the enzyme activity remains the maximum value of enzyme activity remains the same. However, this slope decreases, right? Okay, wait. And in non-competitive one, the enzyme activity totally decreases because it cannot bind to the substrate anymore. It is very important to remember this graph in order to be good at the enzyme chapter and also attempting the MCQs. Like for example, they ask which type of inhibition decreases the enzyme activity. That is the non-competitive one. Moving on, we have uncompetitive inhibitor. This is also important. Sometimes it is not a very frequently asked question, but sometimes they do throw like a curveball or something, and then the students are like, "What is an competitive inhibitor? We have not done that. We do not remember that." Okay, so uncompetitive inhibitor is that, for example, some enzymes have two types of substrate. Okay, now this is the enzyme here. Let me show you. Now this is the enzyme here, and it has two types of substrate, right? These are the two types of substrate that I'm drawing. Please ignore my drawing again. This is the substrate 1. This is the substrate 2. Now, a competitive inhibitor is that which binds to the active site in place of one of the substrate. Now, again, this is the, let me copy this thing, right? Okay, now we have this thing here. Now, one of these substrate binded to the active site, right? But in the place of this one, in the place of this inhibitor, there comes another inhibitor. In place of this substrate, sorry, there comes an inhibitor which is in the shape of this, for example. Now you can see that this can bind to the active site as well, but this cannot make the product now. So that means the enzyme cannot work because it is not the right substrate for the enzyme. One of them is the right substrate, but the another one is not. Okay. Moving on to feedback inhibition. What is feedback inhibition? It is based on end product inhibition. It is also known as end product inhibition. And what does end product inhibition means? It means that, for example, we have this metabolic pathway here, right? A is getting converted to B is getting converted to C is getting converted to D, and these are all the enzymes, right? A, B, and C. The small letter ones are the enzymes. For example, the product is D. The amount of D has increased considerably. Now this product can go and inhibit this enzyme at A, at this pathway. It will inhibit this enzyme A, and the enzyme will stop functioning. A will not be getting converted to B anymore, right? Just because this product D has increased, and this was the inhibitor of this A, and the enzyme A. Okay, that is meant by feedback inhibition. Moving on to the last topic, which is the classification of enzyme. First classification is done on the basis of reaction, and another classification is done on the basis of substrate. On the basis of reaction is we have six type of enzymes here. Uh, First of all, we have oxidoreductase. Let me first do this. We have oxidoreductase. It is used in redox reactions. We have transferase, which is used in transfer of functional group. We have isomerism. We have isomeresis, which is used in isomerism. Hydrolase and lyase is important because it is the breakdown by adding water, and this is the breakdown without adding water. You need to remember this. Okay, ligase, for example, we remember the DNA ligase, which is used to join the Okazaki fragments in the DNA replication on the DNA translation. So we remember that it is used in the joining of compounds. Moving on the basis of substrate protease, they'll act on proteins, right? We all know that. So there are three types of proteases, three or four types of proteases. These we need to remember. For example, pepsin and trypsin has the same function. They are used to convert proteins to peptones. Okay, you need to remember these are peptones, also known as peptides, right? Small polypeptides, these are also known as peptones. Okay, what is the function of carboxypeptase? It converts these peptones or peptides into dipeptides. The function of ellipsin will be to convert these dipeptides into amino acids. You also need to remember these three. These three are actually important in the chapter of digestion as well. Moving on, there is lipase, also known as pancreatic lipase. It converts lipids into fatty acids and glycerol. It converts emulsified lipids, by the way, into fatty acids and glycerol. We have carbohydrates, and the example include amylase, cellulase, sucrase, maltase, and lactase. And then at last we have the nucleases. These include DNAs, RNAs, ATPase. Also, we can see ATPase in that myosin binding in the myosin head. There's ATPase so that the crossover bridges can be destroyed, and then it can come into its original form. So this was all it about enzymes. I hope you guys have enjoyed this session. You guys have understood whatever you guys needed to understand, and it is very helpful for you to also, in order to get the PDF of this whole document, you guys need to comment down below, and then send you a link to the document. Goodbye. Thank you.